welcome to start.
Great. Thank you, Hamid. Thanks for the introduction. And thanks everyone for the invitation to speak. I just want to, acknowledge that, the flood's going on right now. I'm really sorry to hear all that. So, if you're watching this as a recording, don't feel bad. I totally understand. My name's James. I'm a postdoc at the Center for Communicable Disease Dynamics, and I was excited to be able to talk with this group because my interests are really in using medical and epidemiological data to improve public health surveillance. So matching data and methods, which I think is clearly the interest of everyone here. And so specifically, my work is mostly about thinking about how we can use biological assay data, which is kind of historically generated and left in the laboratory, how we can use that data to improve our understanding of infectious disease spread and improve surveillance systems. So I'm going to talk to you today about work we've been doing over the course of the pandemic on the use of viral loads to improve virologic surveillance. And I put these two subheadings in here, icebergs and the Doppler effect. And these are just kind of two analogies that are hopefully going to become kind of clear by the end of the talk. So if you find yourselves nodding off, maybe just play a little game of trying to figure out where these analogies come in. So before I present any of the work, I just want to acknowledge a huge number of people involved in the studies, and notably Lee Kennedy-Schaefer and Brian Cleary, who have co-led the main papers I'm going to be talking about today. And also to thank my supervisor at the time of the work. I've moved groups since then, but Michael Minner was the lead author, the PI on most of these papers, and he now works at EMED as the Chief Scientific Officer. And of course, thanks to all the people involved in generating data and providing input on the studies. And I have no funding to declare or nothing to disclose. So the aim of this talk is to go through this idea of using viral loads as a public health surveillance tool. And to kind of get to that point, I want to go through these three questions. So first, I want to kind of get everyone on the same page of what do we mean when we talk about viral kinetics and viral loads? And through that, why do we even bother thinking about what's going on within the host to understand population dynamics? Then I'm going to talk about one example of how we can use that understanding to improve the way that we deploy tests as a public health tool. And it's just going to be one example. There's obviously multiple use cases, but I'm going to focus on one specific example using pooled testing. And then finally, hopefully through these first two parts, you'll be kind of prepped for this final question, which is, why does the population of distribution of viral loads change over the course of an epidemic? And how can we harness that phenomenon to improve our surveillance systems? And I just want to kind of preface that everything here is developed in the wake of SARS-CoV-2, but these are kind of general principles of viral dynamics. So hopefully these ideas will live on in other systems. Okay, so part one. So to really give you the motivation for this work, I just want to give you an idea of the scale of biological testing that's taking place over the pandemic. And this shouldn't really be a surprise to anyone here, but this plot is just showing you the huge amounts of biological material that's been collected since March 2020. And the kind of take-home point of this plot for the purpose of the talk is each one of these data points gets reported as a binary metric routinely to public health dashboards and reports, etc. So each sample gets tested and it gets reported as a positive and negative. But that's really throwing away a huge amount of information because the vast majority of these tests are PCR, and PCR is inherently a semi-quantitative measurement of biological material in the sample. So what this is kind of showing us is there's real opportunity here to harness a lot more biological information than we currently are. And to kind of get to how we can use that biological information, we need to go under the hood and look at what's happening in the host over the course of an infection. So being shown here is just a really simple schematic of a typical time course of infection for an acute viral infection. So, really simplified and really obvious stuff, you get infected, the amount of virus in your system goes up. Hopefully, as you start to recover, it subsides. Other kind of proxy measurements of amount of virus. So for example, viral RNA follow similar kinetics, but maybe hang around for a little longer, depending on biochemical properties, the thing we're looking for. And then of course we have kind of things related to the host's immune response, which we could also measure, things like antibody titers. So here, IgM and IgG. And even with this simplified schematic, we can start to see why within host biology has a lot of relevance to designing testing strategies. So here showing you how those different windows of time when different within host processes are happening correlate or relates to the time periods when different tests may or may not turn positive. So here we can see that a molecular assay test, so for example, PCR, will turn positive during the period of time when we have elevated viral RNA, whereas an antigen test, which is only looking for viral protein, may turn positive in only a kind of small window of time. So this is just to give us the idea that within host processes are really complicated. And when we take a single sample and test that sample, we're just getting a single snapshot of something very complex going on within the host. And just to make sure we're all on the same page, I just want to spend one slide talking about what PCR does. And I think it's probably all obvious to everyone by this point of the pandemic, but just so we're on the same page, the way that PCR works is that we get a quantitative or at least semi-quantitative measurement called the cycle threshold value. And that cycle threshold value is a measure that's proportional to the amount of virus in a sample or the amount of viral material in a sample. So the way it works is we take a, say, swab sample or a sputum sample or maybe a saliva sample. Through the process of taking that sample, we're capturing bits of the virus. And then we're going to run it through this process of amplification. So the way PCR works is we have an initial amount of viral material, and we see how many times we need to amplify that material until we have a detectable amount of viral RNA. So you can therefore imagine that the fewer amplification cycles we need to get detection, the higher the viral load must have been in the sample. So just to make it very clear for the rest of the talk, a low cycle threshold value corresponds to a high viral load. And so we have our kind of motivation. We see that there's a lot of data we could be doing more with, and we have a simple understanding of how within host processes dictate the performance of diagnostic tests. So now I'm just going to spend a moment talking about a really simple within host kinetics model that's going to underpin most of the work in this talk. So shown here is the time course of an infection within a kind of classic susceptible, exposed, infected, recovered framework. So I might get infected by somebody and go into this exposed compartment. And for a day or two following exposure, I don't have any detectable viral material in my system. Then after a short delay, that viral material starts to increase and peaks at some time, maybe about 5 days post-infection. And if I am going to get symptoms, that's about the time I would have symptom onset. And then hopefully, if my immune system's working properly, I'm going to clear that infection and recover. So all of this is just to say that within host viral kinetics are dynamical, but they're very simple. Viral loads go up and then they go down. And of course, there's variation in the rates at which those things happen, but for the most part, everybody goes through a very similar within host process. So that's great. That's an easy way to model what's going on within one host. But of course, we're interested in thinking about describing population dynamics. So the point I want to make here is that for all one individual is going through this kind of time-varying process, if we consider what's going on across all infected individuals in the population over the course of the epidemic, we see that people have various viral kinetics. And so if we were to take a sample at some point in time in the epidemic, we would be observing some variable distribution of viral loads depending on the stage of people's infections. So this is just to set the scene that at any given point in time, there is a range of viral loads in the infected population. Okay. So just to summarize part one, now that we're all on the same page, key points here are that within host processes are dynamical too. So when we have a single test at a population level, that's a snapshot of a lot of complexity within the host. This obvious point that viral loads go up, then they go down. And then any given point in time, there's a distribution of viral loads in the infected population. So this is to kind of get us to start thinking about what a population of test results is going to tell us about an epidemic. So thinking about testing as a public health tool rather than specifically a clinical diagnostics tool. And just as an aside, it was actually this kind of simple modeling framework that led to this kind of initial work on why regular testing is a good public health strategy and why we can use frequent low sensitivity tests to compensate for high sensitivity tests as a public health intervention. But I'm not going to talk about that today, but I would direct you to this paper by Dan Larimont and Brian Wilder for modeling work on rapid and regular testing. Okay. So part two, now we have our framework of kind of understanding within host processes and how we're going to observe those within host processes through surveillance systems. How is that going to help us deploy testing as a public health tool? And I'm just going to give one example on this idea of addressing resource constraints. So it seems like a distant memory at this point, but you'll recall that throughout 2020, particularly places like the US and lower resource countries, there was a massive shortage of testing capacity. So you can see here articles giving anecdotal data and evidence that people were waiting up to 2 weeks to get a test result back. So I might feel sick, I go to get a test, but I have to wait 2 weeks before I actually find out if I was positive or not. So in terms of testing as a public health tool, this basically makes testing almost useless. If I'm having to wait 2 weeks to get a result, anything could have happened in that 2 week period. I could have gone out and infected however many people. So we wanted to think about how can we use the technology we already have to improve the efficiency with which we use these resources. And I think I've seen on YouTube, you've already had somebody talk about pool testing, but we wanted to think about how we could improve pool testing through the lens of considering distributions of viral load. And so just as, this is historically a very well-established tool for resource saving. So it was actually developed during World War II to test soldiers for syphilis. But overall, the aim of pooling is to use as few resources as possible to process as many samples as possible. So the way it works is that we have a set of samples, we mix them together and we test the pool of samples. So we've used, say, one test to process 10 samples. But there's a potential cost here because through the process of pooling samples, we open ourselves up to potential dilution effects. If I have a salt and water solution and I add more water, it's going to dilute the salt. So it's the same thing here. If I have a sample with some amount of viral load and I mix it with a lot of negative samples, the concentration of viral load in that sample is going to be lower. So it's going to be harder for me to detect it. So we wanted to kind of use our framework of viral load dynamics and viral load distributions to understand how we can optimize pool testing strategies whilst accounting for variation in viral loads over time. So the question of this paper was, what are the optimal pooling strategies for a given epidemic context under different resource constraints? And the way we approached this was through a really simple set of simulations. So the first thing we did was we fitted a hierarchical within host model of viral loads. So literally just that up and down line you've seen on the previous slide. We fitted that to the available time course viral load data we had at the time. So a set of individuals, each individual has their own viral load trajectory that best explains their observed viral loads. So we have that kind of within host model, and we combine that with a simulation of population level dynamics. So simulating an SIR-like model to get a population of varied infection times. We combine those two things, and what that gives us is as big a population as we want of simulated viral load trajectories. So you can imagine now we can simulate the process of going out into this population, taking samples, getting these kind of known viral loads, and running them through in silico pooling strategies. So this slide I'm going to go through quite quickly because I don't think anyone here is looking to set up a pool testing laboratory. But the point is just to show that once we've kind of simulated this population of synthetic viral loads, we can then go in, look exhaustively for optimal pooling strategies under a certain set of resource constraints and depending on the time of the pandemic or the stage of the pandemic. So a lot of obvious things come out here. So one example is if we have more tests available to us than we have samples to process, it always makes sense to just test all samples individually. But as our testing capacity starts getting outstripped by the demand for testing, it starts making sense to do more and more complex pooling. So as you go from top left to bottom right, it makes sense to do more and more complex pooling because the potential savings are much greater. And on the right, it's just showing you just how much efficiency we can gain from doing pooling strategies and also how it depends on the stage of the epidemic. So you can see that at low prevalence, pooling is really, really efficient. And as prevalence starts to get higher, it becomes less efficient. And so I don't want to spend too much time talking about the kind of logistics and resource part of this, but thinking more about the value of considering viral loads in doing this analysis. So because we have a synthetic population where we know which samples we're testing and we know which were positive, we can figure out through our pooling strategies which samples we expect to miss. And unsurprisingly, we tend to miss low viral load samples because those are the ones that get diluted below the limit of detection. But what it turns out is that during the epidemic growth period, 75% of the samples we miss from pooling are those who are more than 7 days post-symptom onset. So those individuals right at the tail end of infection. And of course, this makes sense because we have our viral kinetics model. You can see that right at this tail end here, where people have low viral loads, those are the ones we're missing. And that's kind of reassuring. It means that the people we're missing are those that potentially with least relevance to onward transmission because they're recovering and probably no longer infectious. But what's really interesting is through the simulation is that we observed that this proportion changed depending on the stage of the epidemic or the direction of the epidemic. We found that during the decline phase, this number increased to 96%. So when the epidemic's declining, almost all of the samples we're missing are those at the tail end of infection. So that's reassuring, but it's also curious. Why do we expect this number to change depending on the stage of the epidemic? But great thing, we have our simulation, so we can back and look at the distribution of viral loads over the course of the epidemic. What it turns out is that that distribution of viral loads is changing over time. So what's being shown here is that this distribution of cycle threshold values in our simulated population depends on the time of the epidemic. So at the start of the epidemic, when it's growing, we have a lot of low CT value individuals, high viral loads. And as the epidemic begins to subside, we see this buildup of really high CT, low viral load individuals. So this is telling us that the distribution of viral loads is not just a kind of function of time since infection, but it's also a function of time of the epidemic. And that's going to lead us to our next section. But to summarize this part, what we've seen is that pool testing can be optimized depending on epidemic and resource context. And we can do that optimization much more effectively if we account for viral kinetics. So final part of the talk is kind of answering this latest question, which is why does the population distribution of viral loads change over the course of the epidemic? And this may seem like a kind of theoretical question, but it turned out this is something that was puzzling people throughout the pandemic. And this is quite a recent plot showing you the cycle threshold values from all surveillance testing in South Africa since 2020. And what you can see is this really nice inverse correlation here, where when cases start to go up, cycle threshold values tend to go down. So viral loads start to go up as cases go up. And then as the case curve begins to subside and decline, viral loads also go down, CT values go up. So clearly there's this kind of pattern of cycle thresholds in our surveillance testing systems changing depending on the stage of the epidemic in real life. And the reason this is kind of important to answer is this could lead to all sorts of speculation, all sorts of, so for example, I remember in 2020, an Italian doctor had posted an article claiming that by this phenomenon was suggesting that the virus was becoming less virulent. CT values were increasing, therefore the virus is not causing a severe disease. If that was true, that would be great, but it's quite, fuel for the, for a certain crowd. So we want us to understand why is it, why can we explain this with kind of data science and mathematical modeling? And to answer this question, we can look to demographic theory. And I put these population pyramids to start thinking about why distributions of things change depending on population dynamics. So we have two population pyramids here. So one from Japan, which is a kind of prototypical aging population, and one from Afghanistan, which is a growing population. What you can see, quite obviously, that there's a skew towards older age groups in the aging population and a skew towards younger ages in the growing population. And what this is a reflection of is the differences in birth and death rate of those populations. When we have low birth rates and low death rates, ages tend to increase and build up. Whereas when we have high birth rates and high death rates, we have this kind of aggregation of young individuals. What it turns out, though, is that this is exactly the same phenomenon going on in our CT value distributions. So rather than thinking about ages of people, we can think about ages of infections. Rather than thinking about births, we can think about incidence. And rather than thinking about deaths, we can think about recovery. So shown here is a really simple simulation of an SIR model, where we look at the time since infection or the time of infection distribution at different stages of the epidemic. So let's say at day 75, when the epidemic is growing, we could go out into the population and ask people, when did you get infected? So get a distribution of infection ages. What it would turn out is that there's a kind of skew towards newer infections, so these young infection ages, and very few old infections, because the epidemic hasn't been going on long enough for all these infections to build up and grow old. Whereas if we did the same survey at the tail end of the epidemic, when cases are declining, we would see that most infections are old and there are very few new infections. Again, entirely intuitive, because the incidence curve basically acts like a histogram of the time since infection distribution. Most of the people who are currently infected here were infected at the peak, and every day we go forward, fewer and fewer new people become infected. So that's telling us if we could do such a theoretical survey and find the time-sense infection distribution, we should be able to work out the growth rate of the epidemic. That would be great, but that's impossible to do. People don't know when they get infected. But what we do have from our simple within-host model is this proxy measurement for time-sense infection. We know that cycle threshold values change somewhat predictably, depending on how far into your infection you are. So rather than looking at the time-sense infection, we can look at the distribution of cycle threshold values. And it turns out that these cycle threshold value distributions, therefore, can be used to directly infer the growth rate and direction of the epidemic. So you can see here on the left that when the epidemic is growing, we have this buildup of high viral load individuals. When the epidemic is declining, we have very few high viral load individuals and a lot of low viral load individuals. So that kind of motivated us to go and think, can we use this phenomenon to develop a new surveillance tool? Can we use cycle threshold values to infer incidence, whereas previously we were relying on case counts? So the way we did this, and this is going to be my only kind of heavy methods slide, because I normally talk to public health people. And this is, I'm going to kind of build up the way that our method works. So we start with this idea of we want to model the probability of infection over time, so the incidence curve. And this could be any arbitrary infection curve. It could be the incidence curve from an SIR model. It could be that we estimate incidence as a daily parameter, etc. What we do is we convolute that with the probability of observing CT values for a given number of days since infection. So convolving the probability of infection curve with our viral kinetics curve. And what that gives us is our expected cycle threshold value distribution. So here is just kind of showing you the results of model output and some simulated data. All of this is just to say that if we have probability of infection curve, we have viral kinetics curve, we can generate expected CT distributions. And similarly, if we have any two parts of this three-part equation, we should be able to infer the third. And so here, that's exactly what we're going to do. We have our observed data, CT value distributions. We have knowledge of the within-host process, our viral kinetics model, and we want to infer this probability of infection curve. And I normally, as a rule, try not to put equations in my slide, but for this audience, I thought I should treat you with one equation. And this is the likelihood function that underpins all of this. It's the workhorse of this whole method. So all we need to do is we want to infer the daily probability of infection, pi t, given that we have observed a set of cycle threshold value data for a lot of individuals. And the way that we do that is we go through this convolution process of combining the probability of observing CT values over time, PA, times the probability that people are detectable that many days post-infection, times by the probability that they were infected that many days in the past. And the reason it's really great to write this equation out is we can start to see how we can add an extra complexity to account for real-world variation in surveillance strategies. So for example, we could have an extra term here to account for the way in which people are tested. So maybe people are tested due to symptom onset, in which case we could add an extra probability distribution on in here. So what we do is we implemented this in a Bayesian framework with MCMC. So that allows us to put informative priors on the viral kinetics part of the model. So we don't have to kind of assume a fixed set of viral kinetics. We can put in whatever external data we have available to us at the time. And to kind of cut a long story short, this is what the result of applying this method to cross-sectional CT value distributions is. So this is just showing you a simulation recovery experiment where we simulate a known incidence curve in red. From that incidence curve, we simulate CT value distributions, and we see how well we can recover the incidence curve each week as we add more and more data in. So this is showing you that just using the CT value distributions, we are able to accurately figure out the incidence curve. And what's great is that we don't need to know the proportion positive, we don't need to rely on case counts, we're just using CT value distributions. And to show you that this isn't just something that works in simulation, but indeed works in real life, what we did was we used CT value data, so a set of hospital testing data from a hospital in Massachusetts. So every week there is a new cross-section of CT values, and we feed that to a model, and we're able to accurately reconstruct the incidence curve in Massachusetts over time. And that's great. We can get really complex curve reconstruction if we have multiple cross-sections. What's even cooler is that we can do this with single cross sections as well, right? Just like the age population pyramids are a single point in time measurements, we can do the same thing with CT values. So these dials are kind of showing you the result of fitting models to single cross sections and inferring the growth rate conditional on a single point in time. So a really powerful tool. We can go out into the population at a given point in time and figure out the growth rate of the epidemic without waiting for case counts to build up. And just to show you that this is somewhat reliable, we find that this is really nicely, for Boston, this is really nicely correlated with the wastewater surveillance system. So this is showing you viral loads in wastewater over time. And we see that our reconstructed incidence curves from hospital testing really nicely matches the pattern. Let me just check. Okay, cool, doing good for time. So the final thing to talk about that comes up from this finding is that this actually has implications for interpreting data from SARS-CoV-2 variants. So when new variants have emerged, we've seen increased rates of transmission. And it's important to understand what is the mechanism behind why rates of transmission are changing. Why is a new variant more transmissible than something else? And one of the kind of leading hypotheses, particularly in the first few variants, was perhaps these variants were eliciting infections with higher viral loads, or perhaps infections are more persistent. And support for those ideas was coming from population surveillance data. People were getting their testing data from a hospital or a surveillance system, comparing distributions and saying, okay, this new variant of concern has lower CT values than the old variant. Therefore, it has higher viral loads, and that explains why it's more transmissible. And shown here is one example where that's being done. You can see that as alpha starts to take off, as delta starts to take off, the mean CT values or CQ values here begins to decline, and the old variants CT values tend to be higher. But the reason that our kind of findings from this previous paper are important is that we find that epidemic dynamics are a confounder for this sort of comparison. We can't just naively compare CT value distributions between variants because the underlying epidemic dynamics might be different. And so here you can imagine delta's increasing in incidence, so it has a lot of new infections and therefore high viral loads, whereas alpha is declining in incidence. So all the infections the samples we're getting are from old infections, so typically lower viral loads. So this is pointing to our next aim, which is we want to understand how can we account for these differences in dynamics when comparing viral loads across variants. So to get at this, and I'm just going to show a couple of kind of first few results from this paper. We simulated, made a really simple simulation of a 2-strain SIR model. So here we have the original variant with some kind of low level of transmissibility getting replaced by a new variant, which is much more transmissible. Right, so it's 2-wave epidemic representing 2 variants. Then we kind of overlaid a viral kinetics model, and we kind of simulated 2 scenarios. One where the new variant has identical kinetics to the old variant. So here, this kind of blue and green line here. And also a different scenario where the new variant does indeed have more higher viral loads and more prolonged infectivity. So this orange line here. What we can see is that if we simulate both scenarios, even when the viral kinetics of the 2 variants are identical, if we went out into the population at a time when they have opposing trajectories, we would find that comparing the blue line to the green line would lead us to make an incorrect conclusion that the new variant has higher viral loads. So what this means is that we need to figure out a way to account for growth rates or account for epidemic trajectory when comparing viral loads between variants obtained from surveillance samples. And I'm not going to talk about that, it seems. So I would direct you to the preprint here if you're interested in hearing more about that. So to summarize this final section, viral load distributions are predictable from epidemic dynamics and viral kinetics. We've shown how we can use this information to estimate incidence using single cross sections of detectable CT values. And we propose that this is a potential tool in settings with limited test capacity because we're not having to wait for this buildup of a time series of cases to infer incidence. We can use single cross sections of data and infer epidemic trajectories. The other great benefit of this sort of method is that we're using information in distributions, not in counts. So you can see here the scale up of testing over time in the US. And what this presents a problem for case count-based incidence estimates, because we can't necessarily know if we're seeing more cases because more cases are occurring, or because we're just simply testing for more cases. This method doesn't care about that because we only care about the distribution of cycle threshold values. And the final word of caution on comparing within those quantities between populations which have different dynamics. Okay. So to go back to our initial analogies, so I like to think of viral load distributions in populations as a bit like an iceberg. And what we've shown is that epidemic dynamics dictate both the position and the shape of that iceberg. So what that means is that depending on what the test we use, we're going to see different parts of the iceberg, right? So if I have a more sensitive test, my my limit of detection goes down. I can detect smaller amounts of virus. So this kind of line of water will drop. Whereas if I have a less sensitive test, my limit of detection goes up and I see less of the iceberg. And what we've shown is that the shape of that iceberg and what's above or below the water depends on epidemic context. And I think kind of a lesson relevant to maybe people in the AI and machine learning world is biomarkers are not static things. They do may or may not reflect context within those dynamics. We also have to think about the context in which we obtain distributions of biomarkers. So I also want to kind of use this as a way to think about how we can extract more information from existing surveillance data. So rather than asking for the generation of more data, is there kind of information inherent in data we're already collecting that's currently getting discarded? So here, viral load distribution. So thinking about how we can use tests with their full quantitative value rather than reducing them down to binary metrics. And also thinking about how training set distributions are important depending on epidemic dynamics, and not just for kind of statistical analyses, but this is an important finding for regulatory assessment of diagnostics and surveillance tests. So if I want to kind of have a panel of samples to see if my test works, I need to think about the distribution of those measurements for assessing whether my test is sensitive or not. And that's all I have. So thank you everyone for your attention and happy to take questions.
Right. Thanks, James. It was a great talk. So we have like 10 minutes for the questions, but before questions, I would say that as you may know, many of us are affected by the Queensland rainfall and flooding in the past few days. So I can see that most of our colleagues couldn't join today, but I'm sure they are looking forward to catching up with your talk on YouTube. So, okay, we have like 10 minutes. I can see Ash has posted 2 questions in the chat. Is the threshold comparable across? Ash, do you want to explain which threshold you are referring to? All right, maybe.
So I can take a stab at interpreting the question. So
yeah, maybe. Okay, yeah.
So thresholds, I'm seeing from timestamp this was at the start. So this is, I think, the threshold of the cycle threshold, so this amplification process. So I guess it's less pertinent to think about people and more pertinent to think about the technology and the assay being used. So different machines have different calibrations. They have different thresholds. So a number on one machine doesn't necessarily correspond to the same thing on a different machine. So that's again another caution for comparing quantitative data generated from surveillance systems, which may use lots of different technologies. We need to think about standardization. But in general, you know, with the measure, the instrument is looking at the sample, and once you have the sample, it doesn't necessarily care who that's come from, right? Of course, there are predictors of kind of viral loads during the course of infection. Things like age does have a small impact, whether you've been vaccinated or not, whether it's a so-called breakthrough infection, that sort of thing. So I've really simplified the dynamics that we're considering when generating viral load distributions. But more complex versions of this model and this approach do need to think about things like the demography of the people that are getting infected and the immune status of the people getting infected.
Any other questions before going to the chat menu?
Hi, James.
Okay.
So just reading your question on.
Yeah.
Do you want to?
James.
Yeah. Sorry for gate crashing a YouTube meeting, but Alexandra, who I think you know, James, alerted us to this talk, and we thought we'd join. We attempted to try to replicate some of this stuff in New South Wales, but getting access to CT values is harder than it would seem. But I'm just curious about. Obviously, there's changes in viral dynamics with the new variants emerging. I mean, not major changes, I suppose, in the fundamental distribution, but different heights and speeds at which things rise. Has that posed any challenges? And I guess maybe it's not helpful, but can you use CT surveillance to detect emergence of new variants? Maybe you don't want to because genomics is already there, but I'm just curious about these sort of features.
Yeah, good questions. Yeah, I mean, I think in terms of understanding population distributions, what this means for surveillance, yeah, we have seen a little bit of evidence that viral kinetics have changed between variants, but not much, right? And I was kind of talking about using population data and surveillance data to figure out if different variants have different kinetics, but really the best data sources are still longitudinal cohort studies, so tracking people from the point of infection and just plotting their CT values over time. And so there's a lot of data being generated on that, and it really doesn't look like variants are super different. You know, maybe Delta has higher peak viral loads by a little bit, but not a whole lot. You know, a little bit of evidence that it differs depending on whether you're immune or not. So, you know, some evidence that if you have a breakthrough infection, you've been vaccinated, your viral loads drop a bit quicker. And that is a bit of a complicator if we want to use these models to estimate things from populations of CT distributions. So as I said, like we developed this method at a time when there wasn't much immunity and there was only one variant circulating. Once you have like mixture distributions of lineages with different growth rates, different kinetics, affecting immune people and not immune people, that does complicate things. And so that has opened a lot of interesting questions, you know, particularly been looking at UK surveillance data, so kind of routine like symptom-based surveillance data. And we do see really clear time-varying patterns, but they don't match up just to periods of growth and decline of cases, like in the South Wales case. And I think this is showing us that the distribution of viral loads is really this complex mashup of all these kind of contributors.
Yeah, no, thanks. I was just curious because obviously influenza is another thing you might consider doing this for, which has all those features you're talking about.
Yeah, absolutely. I'm very keen to see how these will work for other pathogens. I think it's going to depend on how much external data and studies we have on within-host kinetics, right? And I think for something like flu, we do have a lot of kind of past work on figuring out how viral loads go up and they go down over time. So I do have hope that this will work for something like flu.
Other questions? If not, maybe the second question from Ash. Does the viral load or the threshold correlates with any external symptom like cough, sound?
Yeah, so again, not as much as you might expect. Like it's very intuitive to think if there's more virus, you're going to get sicker, but the data doesn't seem to support that. It seems to look, if you've got infected, there's enough virus to kind of make you have a detectable infection. Your viral load is going to go up and go down at pretty much the same rate. Maybe there's a little bit of evidence, maybe symptomatic individuals have slightly more persistent viral loads, so they have elevated viral load slightly longer, but again, not by much. Because we really need to think about, you know, symptoms are a function of the immune response. They're not a function of how much virus there is. And, you know, immune response is complex. It's not this kind of dose-dependent feedback. It's you get the immune system detects virus, it sets off a load of cascades, it turns on symptoms. So it's really not kind of as easy to model quantitatively.
All right. Any other questions?
No, not silly at all, Ash. Thanks.
All right. If there is no other questions, thanks very much again, James. and participants for joining us, and looking forward to see you in our next seminar series. Thank you.
Yeah, thanks everyone.
Thanks very much, James.
Thanks. See ya.
Thank you.